Pathology Fellow, finishing up her training at NYU Langone Health. She completed her APCP residency at SUNY Upstate Medical University and attended medical school at Ross University. She was most recently a research scientist in Mattia Snuderl's lab at NYU Langone Health. Kristyn finished neuropathology fellowship in 2022. She will begin a faculty position at University of Washington in Seattle in August of 2024. And I just want to say I am really looking forward to this talk. I am really looking forward to it.
Thank you. So thank you to AANP for allowing me to discuss DNA methylation with you today. And I can't progress. There we go. I have nothing to disclose. These are the learning objectives for the lesson today. And so this is what we're going to be talking about. So basically, we're going to start with what is DNA methylation? How was the classifier developed? How do we use the classifier in tumor diagnostics? When should we use the classifier? When do we get the most utility? How has DNA methylation been included in our WHO guidelines? What are the diagnostic challenges that we see when we use DNA methylation in our practice? What are the future uses of DNA methylation in tumor diagnostics? And then, of course, how do we pay for it? So first, what is DNA methylation? DNA methylation is the addition of a methyl group to DNA. This occurs at CpG sites throughout the genome. It's an ancient evolutionary epigenetic modification that's involved in chromatin structure, gene silencing, and genetic stability. This methyl mark is maintained throughout cell divisions, establishing an epigenetic mark of the genome. The genome-wide DNA methylation pattern is a composite of methylation patterns of the cell of origin, in addition to changes that are accrued due to aging, environment, or mutations. Methylation of patterns of tumors remains preserved and accurately reflects the cell of origin. It remains stable throughout the course of disease, and it can serve as an epigenetic fingerprint that we can use for tumor classification. So DNA methylation can be analyzed using many different platforms. Whole genome bisulfite sequencing is the most comprehensive, but it requires very high quality DNA. It's very expensive, and we can't use it on formalin-fixed paraffin-embedded tissue, so it's much less useful in our clinical practice. Targeted bisulfite sequencing is also not compatible with FFPE tissue. And then DNA methylation array, which is what we'll be primarily discussing today, is FFPE compatible. It's an immensely robust assay. It requires a relatively low starting DNA input and has a lower cost than other methods. So I feel like DNA methylation is kind of a black box. We put tumor in and then we get an answer out of it. And so we're going to kind of open that black box and go into the nitty-gritty of a little bit of the technical aspects. And so DNA is extracted using any method of DNA isolation. Remember I said that DNA is methylated at CpG sites throughout the genome. So DNA undergoes bisulfite conversion. And what it does is that any C that is methylated stays a C, but any C that is unmethylated is converted to a U. Basically, the DNA methylation is a bead chip array. So DNA from the tumor is added to a bead chip. On each chip is about 8 chambers, allowing for 8 samples to be tested per chip. In addition to the tumor being added to the chamber, you have individual beads that are added, and these beads have a barcode that identifies the physical location on the bead chip, as well as a 50 base pair probe. And these probes are specific for the methylated sequence and the unmethylated sequence. So these probes are designed to be complementary to specific 50 base regions of bisulfite converted DNA. After hybridization of the DNA to the probes, a single base extension of the probe incorporates a fluorescently labeled marker. So this bead chip is in a machine called the iScan. The iScan essentially takes a picture, which is an IDAT file. This is the raw file. This IDAT file is then processed through customized bioinformatics pipelines. And what it does is it reads the intensity data, or what the bioinformatics does, it goes through the intensity data for each probe. And so the proportion of DNA methylation at each CpG site is converted into a ratio of methylated to unmethylated. It's like methylated being 1 and unmethylated being 0. And this is known as the beta value, and this becomes important in a couple slides. So just a quick overview of what I just said. Tumor is added to the bead chip. The bead chip is run through the eye scan, and this presents this picture of kind of red versus green that is then bioinformatically deduced into a beta value. So how did they develop the classifier? First, there was a established reference cohort of CNS tumors, and this included our WHO-defined tumors, as well as additional tumors that we see in the CNS, such as mesenchymal tumors, melanoma, and our lymphoid tumors like diffuse large cell B-cell lymphoma and plasmacytomas. These tumors underwent genome-wide DNA methylation. They did unsupervised clustering performed on within each entity and then across histologically similar entities. And this resulted in 29 classes that match a single WHO entity, 29 classes that were a subclass within a WHO entity, 11 classes that were not identical to a WHO entity, and then 5 novel classes that were not defined by the WHO. This is highlighted on the right just to show you the different tumor classes that were involved and then how they would cluster on a T-SNE, which we'll get to in a couple slides. So then basically a machine learning random forest algorithm was employed, and this generates 10,000 binary decision trees with the data from all of the reference samples. And so looking at each methylation sites, looking at the beta value of methylated versus unmethylated, you follow the decision trees down until you get to a single entity. Each tree assigns a given tumor sample to one of the 91 classes, resulting in this aggregate raw score. And then this raw score is transformed into a probability that measures the confidence of the class assignment. And so this is what we call the calibrated score. So again, you have the tumor of interest, you run it through the DNA methylation array, you compare it to a reference cohort, and then you get a result of how your tumor classifies. So this is what our report looks like. You have the classifier results up here. We focus on the family, which would be diffuse glioma IDH mutant, a calibrated score of 0.985, and then the subclass, which is astrocytoma IDH mutant lower grade. And then how we evaluate the calibrated score is as follows. So above 0.9, we call positive. Between 0.3 and 0.9 is indeterminate, but may still be informative. And then less than 0.3 is negative. We can't do anything with that class. There's a T-SNE, which is a statistical 2D representation of data that shows you where your tumor is. So you can see where this tumor classified in relation to the reference cohort. And then in addition, we see a copy number plot in our report, and this is done using a Economy package in R. And then how we implement this in clinical practice is the most important thing, and many studies have been published talking about their experience. Heidelberg, who developed the classifier, published on their experience in the classifier. The NCI published on their experience as well. Both of these came from referral institutions and had kind of an inherent bias towards these difficult to diagnose cases. So when we were presenting on methylation, we were often approached by people who asked us, like, we know we should do it on the weird things, but when else should we do it? Should we do it on everything? And so this led us to at NYU to, which is a more representative of a clinical practice, not a referral institution, to publish our findings and our experience with the DNA methylation classifier. So we did a prospective study to propose guidelines for the use of DNA methylation in routine clinical practice. We don't currently have guidelines for when we should use DNA methylation. So we looked at all of our tumors diagnosed between 2014 and 2021, with 1,921 brain tumors undergoing DNA methylation. All tumors received the standard of care pathology diagnosis at the time of review, as well as simultaneous whole genome DNA methylation profiling. I know this is a busy slide, but I'll walk you through it. Our cohort, you can see in the pie chart on the left here, consists of 1,602 WHO recognized diagnoses and 319 descriptive diagnoses. So these include our low-grade glioneuronal tumor, low-grade glioma, high-grade glioma, PNET, et cetera. Just looking at the WHO diagnoses alone, we saw that 74% of these were a diagnostic match between histology and DNA methylation. So the histologic diagnosis and the DNA methylation diagnosis were concordant. About 14% of these were discordant. So there was DNA methylation and histology did not match. 7%, about 110 cases, received additional prognostic significance, so our ependymomas and our medulloblastomas, and about 5% of cases were unclassifiable by DNA methylation. We then looked at our descriptive diagnoses and found that DNA methylation was able to give a diagnosis in 86% of cases, allowing for a clinical discussion on treatment that was more accurate than our descriptive diagnoses could provide. The other thing that I would really like to highlight is that people again asked us, when do we use this? And I think a take-home message from our paper is that DNA methylation is more useful in our clinical practice than just the difficult to diagnose cases. I think that most of us would say that we're relatively comfortable diagnosing glioblastoma histologically. We actually found that, and just to orient you to the slide, the dark blue corresponds to a complete match. The light blue corresponds to a diagnostic mismatch. And then this kind of intermediate blue is cases that were unclassifiable. So in our glioblastoma, 82% matched as a glioblastoma, but about 13% of cases matched as something else, and kind of provides the utility for DNA methylation in these cases. In addition, it's incredibly useful for our glioneuronal tumors, which are notoriously difficult to diagnose by histology alone. And then just in general, it's useful for prognostication of our ependymomas and our medulloblastomas. And also, I'm sorry, the Sankey plot on the right shows the histologic diagnosis on the left, being separated into the different DNA methylation diagnoses on the right. So moving on from our WHO recognized cases, we then analyzed our descriptive cases. So we had 319 descriptive cases, and we split these into adult and pediatric cases, and then high-grade versus low-grade cases. And the take-home message from this slide is that everything that is not in dark blue was able to be given a diagnosis. So 86% of descriptive diagnoses were able to be classified by DNA methylation, increasing the accuracy of our diagnosis. So this led to our proposed criteria for the use of DNA methylation in clinical practice, and we stratified these into high yield, intermediate yield, and low yield. And our high yield is obviously CNS tumors that are defined by DNA methylation signatures. So these are things like our HGAP and our DGONC. All CNS tumors that we can't give a WHO diagnosis, and so we give a descriptive diagnosis. Tumor entities with a high chance of diagnostic error in the absence of other molecular studies, like our pediatric low-grade gliomas or our glioneuronal tumors. Tumors with inconclusive or contradictory immunohistochemical or molecular results, and the tumor where subclassification may affect clinical management or provides prognostic information, again, like our medulloblastomas and our ependymomas. Our intermediate yield are cases in which tumors in which DNA methylation could triage further molecular testing, so maybe help us decide between DNA and RNA NGS. Tumors with moderate chance of diagnostic error in the absence of other molecular studies, and then tumors in which greater than 10 immunohistochemical stains and or multiple molecular tests may be required for diagnosis. This could help in tissue preservation and cost efficiency. And then our low yield are tumors with low chance of diagnostic error when using recommended techniques according to WHO criteria. And so these are things like pituitary adenoma, schwannoma, meningioma, and then also tumors in which other molecular tests have already established a molecular driver in a tumor classification, and then tumors where there's no established prognostic value of molecular subclassification. So this is how the DNA methylation classifier was developed. This is how we think it should be used in clinical practice. And so the next step is, well, how has it been incorporated into the WHO? So obviously there are DNA methylation defined entities. These are the high-grade astrocytoma with pyloid features and the diffuse glioneuronal tumor with oligodendroglioma-like features and nuclear clusters, which I prefer to just refer to as DGONC. And And then entities in which DNA methylation has been included in the diagnostic criteria. And this is an example on the right in those boxes in the WHO that have the diagnostic criteria. This is for diffuse midline glioma, K27 altered. You have your essential criteria, which is your diffuse glioma, K27 loss, midline location. And then, for where you could, it's any of these, and methylation profile is one of the things that's been included. And this is common for many of the different entities in the WHO. So once we have used the DNA methylation classifier in clinical practice, we come across these challenging scenarios, and kind of the most common challenging scenarios that we see are that the tumor does not classify with any class and it has a score of less than 0.3. Tumor classifies with a class that makes sense histologically, but with an indeterminate score of between 0.3 and 0.9. And then a tumor classifies with the DNA methylation class that you weren't even considering based on the histology, also with an indeterminate score between 0.3 and 0.9. So then how should we approach these cases? Obviously, first you would like to evaluate for lab error, make sure there's no tumor swap, contamination, et cetera. You could review the H&E for tumor purity. DNA methylation is a robust assay with a tumor content of, we like to say, 50% or higher. An infiltrating edge with a lot of normal can dilute the tumor content. This is obviously true for any molecular assay. Inflammation can also cause dilution of the tumor content. Necrosis without inflammation, we have not seen to have a strong impact on the DNA methylation assay. And then this may spur you to do additional molecular testing, and I'll explain this further. So when we were going through all of this DNA methylation data to come up with some clinical guidelines, we noticed that a lot of our diagnostically challenging cases or cases that did not classify well had alterations for which the classifier was not trained, and that this can affect the classifier result. So this kind of spurred us to look at a cohort of 219 CNS tumors that were positive for fusion by RNA sequencing that also had concurrent DNA methylation. These are cases from NYU, Cornell, MSK, and the NIH. And these consisted of tumors that did not have disease-defining fusions. So this excluded like a pilocytic with the classic BRAF KIA 1549. And what we found, one consistent with other papers is that you can see in B here that we have this circus plot showing the different partners that occur with NTRK 1, 2, and 3. You can see that it has many different partners, and then the Sankey below shows that this is the histologic diagnosis on the left and the DNA methylation diagnosis on the right. There's many different tumors that this NTRK fusions occur in. And then also BRAF was very similar, it had many different partners that occurred across many different tumors. Our EGFR fusions and our MET fusions tended to have partners also only on chromosome 7, and they also primarily occurred in GBMs. And we found that looking at the fusions and how they affect the classifier result, that there were kind of two main ways that this could happen. Basically, the diagnosis by histology and DNA methylation could be concordant, but with an indeterminate score below 0.9, or the diagnosis was completely discordant between histology and DNA methylation. Additionally, this is more just like a fun fact, I think we classically think of tumors as being driven by a single fusion. We actually identified multiple tumors that had multiple concurrent fusions. So the tumor on the left here was diagnosed histologically as a low-grade neuroepithelial tumor, so a descriptive diagnosis, and classified poorly as a DNET with a score of 0.43. You can see that there's chromothripsis of chromosome 7, so this kind of shattering of the 7 with many breakpoints, and that there are actually 3 unique fusions that are involving 6 different genes, all on chromosome 7. And the same thing on the right, this was diagnosed by histology and by DNA methylation. So this was a concordant diagnosis with a calibrated score of 0.89, which is below 0.9, but not so far below that we're concerned about it. But when we did, when we looked at the copy number output from the DNA methylation classifier, again, you can see shattering of chromosome 7 with multiple breaks. And again, 3 unique fusions involving 6 genes all on chromosome 7. So we think these additional alterations are actually affecting the classifier result. And these show the two ways that it happens. You have a complete discrepancy between the histologic and DNA methylation diagnosis with a low score. You have a concordant histologic and DNA methylation diagnosis, but with a low score. So these are just some real life examples of challenging cases that we saw as we went through our DNA methylation cohort. So this is a 76-year-old male. He had a past medical history of well-controlled HIV, prostate adenocarcinoma, and adrenal insufficiency. He presented with 5 days of a right-sided headache with accompanying right eye pain and tearing. And then imaging showed this infiltrative, expansile, heterogeneously enhancing mass in the anterior temporal lobe, and a subtotal surgical resection was performed. Histologically, this was diagnosed as a glioblastoma. We did DNA methylation, which showed it, again, it classified as a glioblastoma with an indeterminate score of 0.5. So then this made us look further into what could possibly be going on with the case. This classified by on the t-SNE as a GBM mesenchymal subtype, which is what it classified with on the classifier. We looked at the copy number plot, which showed alterations that are consistent with the GBM, so our EGFR amplification, MDM4, MDM2, and CDK4 amplification. And so we were comfortable based on this, the additional information, that this is a GBM. And so how we would suggest going through this case would be reviewing the t-SNE to see if it clusters with the same class, review the copy number plot to see if there are the expected alterations that you would see in this tumor, and then review the tumor cellularity of the slide for DNA methylation to see if this can account for the lower than expected score. So our second case is a 24-year-old female with a syncopal episode directly after cutting her hand in the kitchen. She had follow-up at her primary care physician that showed evidence of vertical nystagmus. An MRI scan demonstrated a tumor in the left cerebellum and cerebellopontine angle. You can see it was diagnosed descriptively as a pediatric diffuse glioma low grade. It classified very poorly as a low-grade glioma rosette-forming glioneuronal tumor. It clustered on the t-SNE with a completely different class of low-grade glioma, pilocytic astrocytoma posterior fossa. And when we did additional molecular testing, we saw that this had an NTRK2 fusion, and we think this accounts for the poor score in classification. So basically, the take-home is that this fusions can cause a low score matched with an expected class or an unexpected class. So kind of changing gears completely, one of the most common questions that we got, besides, when do we use it and what do we do with the challenging cases, is how do we pay for it? Obviously, this is an evolving landscape. Current approaches include using a descriptive code or codes for MGMT or copy number analysis. The process is underway to establish a specific CPT code dedicated to the DNA methylation classifier, and hopefully more to come on that in the future. And then, what our additional uses for DNA methylation and what are future uses for DNA methylation. So the sarcoma classifier has been published and is in use. Other classifiers are in development, like the kidney classifier, the derm classifier, cancer of unknown primary, and the lymphoma classifier. And then additional uses that we use it for now are MGMT promoter methylation, copy number analysis, and MLH1 promoter methylation. These are my references. I'd obviously like to thank the team at NYU, our collaborators, and our funding sources. And then, thank you for your attention. And if you have any questions, you can take them now.
Yes, thank you, Kristyn. That was awesome. So at this time, we'll move into Q&A. You can submit your questions via the chat or unmute to ask a question. When submitting a question via the chat, please ensure the message is to everyone so that the presenters and attendees are able to see the question. To unmute, select the microphone in the lower left corner. And once you've asked your question, please remute to avoid background noise. We'll give people a minute to ask questions. I have a couple myself, but I would prefer to have the attendees ask their questions. Well, I'm going to go ahead with some of,
Question.
oh yeah, no, please go ahead. Go ahead. Dr. Kazmi, please go ahead.
Yeah, I work at the Geisinger Medical Center, and I was wondering, is there any and are your lab is available for like the outside cases, or is a commercial lab available to send these cases to?
Right. So the question is, kind of where can you send this if you don't have it in-house, essentially, right? So there are, you can obviously send it as a consult to any of the institutions that are providing it. NYU is providing DNA methylation. Northwestern has DNA methylation. I know the NIH accepts cases. I don't believe there's any commercial lab that has that set up, but there are many institutions that accept consults.
My question is, you started with discussing the CPG sites. We all know that's like super important for analyzing DNA methylation, but do the CPG sites or islands, do they have any other use other than us being able to do DNA methylation studies?
I mean, I guess in short, I don't know. The CPG sites are obviously are where the methylation exists. They play a role in like the gene silencing and what genes are expressed. As far as like utility, other than providing a pattern for us to use to develop a classifier, I'm not sure.
Okay, and then so the other question I have is, your discussion, obviously it's, this is a brain tumor talk, but what happens if you run your chip and like it's an adenocarcinoma?
Yeah.
And the clinician on it classified, what do you do at that point?
Yeah, so when we run a case that ends up not being a brain tumor, they end up classifying on the CNS classifier with a poor score, either with like a meningioma or a choroid plexus carcinoma. For some reason with those classes, they tend to sometimes EFT chick as well. And what we can do at that point, I mean, there are other classifiers in development, like the cancer of unknown primary classifier. Those are obviously not ready for clinical use yet, but they're in development. And so we run the tumor through the cancer of unknown primary classifier. The other thing that we had some interesting cases, like we had a lung adenocarcinoma that was run through the CNS tumor classifier, and it classified with like an ATRT with like not a terrible score. Obviously clinically made absolutely no sense for that tumor, but the case had like a SMARC B. It was diagnosed by the lung pathologist as a SMARC B1 deficient, poorly differentiated, like whatever their entity is in lung, like poorly differentiated lung adenocarcinoma. And so we actually do see that sometimes tumors that are not brain primaries but have similar molecular alterations can classify like with an indeterminate score with the CNS tumor with a similar alteration.
Okay, thank you. Then I think I'm gonna go through some of the questions. It says, how accurate is MGMT by methylation profiling?
That's
Victor.
We're actually currently validating this for us. We do MGMT by power sequencing, and we want to kind of streamline our testing because we do DNA methylation on every single brain tumor. So if we can do MGMT analysis by DNA methylation, it just helps streamline our molecular testing. And we actually have found, and there's a great paper for the person who kind of developed the MGMT portion of the classifier that discusses how the MGMT, like by power sequencing, compares like how the sites that you pick compares between the MGMT classifier and other entities. And we found that the MGMT by DNA methylation is more accurate than our power sequencing.
Okay. So I'm going to go ahead with this. Mark Cohen says, many thanks for a brilliant presentation. And Ryan Rebbe says, did you find anything native to the CNS that prevented or hindered extraction or enrichment?
I'm sorry, I don't understand.
I think they're just, I think he means DNA extraction. Like, is it hard to extract DNA? I think you can unmute if,
no.
Compared to other tissues, yeah.
No, I not that I've heard our lab complain about comparative to other analysis. I think the DNA extraction we found to be straightforward.
Okay. And then, great talk. Tab says, great talk, thank you. What do you do when the methylation profile conflicts with NGS data? For example, when a tumor clusters as an adult type IDH mutant diffuse glioma, but NGS fails to demonstrate IDH mutations?
Yeah, that's a really great question. And I think that, we often, DNA methylation is a lab test and it requires interpretation. I think I would just, make sure that your panel is covering IDH, the other IDH mutations that could be possible. I think there's also maybe other ways that IDH could be affected. I think that's a pretty rare occurrence in our history of DNA methylation. I would be either suspicious of a swap, so I'd go down that route. If I found out that the tumor is definitely the tumor that I expect it to be, that I would look at the NGS analysis that was done and make sure that you're covering the other IDH mutations that are possible. And then at that point, I would look at the other things like ATRX, TP53, and see what else we see. I would use other analysis to help me with my diagnosis.
And then Michael Farrell says, what is your turnaround time for methylation profile result? Have you found the meningioma classifier to be of clinical use?
So I'll answer the second one first. We don't use the meningioma classifier specifically, so I don't have experience with it on its own. The DNA methylation analysis itself is a 4-day process from DNA extraction to classifier result. So we run a week, usually 2 chips, and so it's 1 to 2 weeks depending on when we get the tumor sample and how it coordinates with when we run the run.
Sure. Okay, and then Greg Wells, what are obstacles to combining the several available methylation classifiers into a single super classifier? He has other questions too. And then does the methylation classifier undergo retraining as more cases become available? Or on the other hand, is a classifier trained once and then frozen in time?
No, I think the classifier has been retrained a couple times at this point. I think it's more, so there's like version 12 point something right now. And you can kind of see that on the Heidelberg website that they have. But so simple answer to that is no, it's definitely been retrained multiple times. And then, sorry, the other questions were?
I think you answered, what are obstacles to combining them into a single super classifier?
So I don't know enough about machine learning to answer that question. I think it depends. I think the classifiers that are, like the NIH classifier versus the DKFZ classifier, I think the NIH is using a different machine learning algorithm, but definitely don't quote me on that. We only use the DKFZ classifier. And as far as combining it into one super classifier, I'm not sure the limitations there.
Okay. And then Elizabeth Rushing says, excellent presentation. Are you also using nanopore sequencing?
We are not, no.
Okay. Okay, I don't see any more questions. I hope I didn't miss any. If anyone wants to unmute. Okay. We got one more. Greg Wells, do you foresee transcriptomics contributing to diagnosis?
Sure. Yeah, definitely. I mean, yeah, I think it's always important to figure out like how the alterations that we see are affecting expression. So I definitely see that being part of diagnostics.
Are we anywhere close to that currently? Do you know what research explorations in that direction?
I think many people are exploring it. I don't know how close we are to having it be included in guidelines, but I think it's heavily being researched. Well, thank you so much for your attention and for allowing me to talk about methylation.
Thank you so much.
Thank you. Just a few closing announcements. Thank you to everyone for joining today's AANP Teaching Rounds presentation. We would ask that you take a few minutes to complete a short evaluation. Completion helps ensure accurate reporting to the accreditation board. A link to the evaluation will appear on your Path LMS screen upon conclusion of the Zoom meeting, or you can navigate back to the main course and select the evaluation option. Once you've completed the evaluation and selected your credit amount, please then select the appropriate certificate based on your credentials. The PowerPoint slides and recording will be posted to the AANP website within the next week. Thank you again to Dr. Galbraith for an excellent presentation. This concludes the session for today. Thank you, everyone.
Bye, everybody.